Welcome back to Bioinformatics with Baby channel. My name is Babajan Banaganapalli and I'm an associate professor in bioinformatics. On this channel, we will cover various bioinformatics or biological data analysis concepts in genomic, transcriptomics, proteomics, and metabolomics. So if you like this content, please subscribe our channel. Today we are going to talk about some computational methods that are helpful to annotate the protein sequences. So annotation in the sense means we can carry out numerous kinds of analysis like to determine the function, to determine the domain, to determine the transmembrane region, secondary structure, or solvent accessibility. Many more things we can do by these annotations. So before I start into the actual topic, I just want to give us some basic introduction to it. For example, if you want to know why we need to know the function of the protein. So after getting the sequence of the protein from the machine, the next thing is we have to characterize. It means we have to find what exactly the function of that protein. Okay, so generally a good way to study the function of a protein is exactly we need to find out what exactly it happened, what exactly is going to happen when it is absent in the cell. So for this, most of the researchers or most of the scientists use the model system approach. For example, cell culture or whole organism. Okay, so they carried out as they use them as a model system where they can test the function of the specific protein or a gene by knocking out that particular gene or modifying that gene or protein to understand what exactly the phenotypes it will generate during its absence or during its mutations. Okay, these can be revolved with this can be done by using the various experimental method like starting from blotting techniques, and also you can see the more advanced technique like microarray, RNA-seq, SAGE methods. Okay, so in situ hybridization methods. There are various methods are there which can be solved, which can be resolve the structure function of a protein. As I told you in a previous video also, doing the experimental things, it's not as simple as we are getting in the computational methods. It need to have so much of resources are needed, huge funds are needed, huge amount of manpower is needed to do this analysis. Okay, so and also the percentage of success is very low in especially the proteomics technologies to determine the function of a protein. So the alternate method is, we already know that one, the computational method is the very good alternate source for us to understand or to determine the what exactly the function of that protein or what are the domains are there, what are the transmembrane regions are there. Okay, so we can do so many things. Here I gave only 4 or 5 examples for you. So depending upon the type of annotation you required, some thousands of some hundreds of thousands of databases are available or softwares are available. You can do your prediction by using those tools. Okay, so by taking this one, so as I told you, today we are going to look onto the various protein sequence annotation tool. From this tool we can predict the function, we can identify the physical chemical properties of that protein domains. We're going to identify the domain, conserved regions, transmembrane regions, active site, secondary structural element, solvent accessibility, and mutation effects. So one thing you have to remember, we are here talking about the amino acid sequence, not the structure. So we are going to use a single letter amino acid sequence to do all this annotation. So here in this video, I'm going to use top cited and most frequently used tools to perform the various protein sequence annotations. Okay, so let us start. First tool, we are going to check for the physicochemical properties of a protein. So majorly we are going to check by using the protoform XPASI tool. It's one of the best tool to compute the various physicochemical properties such as molecular weight, theoretical pi, okay, central point, amino acid and atomic composition, extinction coefficient, estimated half-life, instability index. All these things we can easily understand by using this XPASI protoform. And one more database is there, almost it is identical with that one. It's called as a pepdra. Here you can draw a peptide. Okay, so in two-dimensionally you can draw a peptide by using this pepdra. So we are going to check these two database, how exactly they are going to work. Now we are going to open them. Let us see this one. Okay, so just we have type here proto pram. Okay, so here proto pram. Okay, so here, so protopalm, this is a tool name. So here it's been protein parameter tool. Okay, protein parameter tool is the first one. So click this one. So already I have a sequence. Okay, so I'm going to use the same sequence here. Okay, so here my sequence name is C1S complementary subunit 1. Okay, so here I'm going to use the same sequence. Okay, you can get this sequence from anywhere. For example, if you want the same sequence, you can get it from NCBI, CAG, Ensemble, Uniport. Okay, so from many databases, you can Swissport, you can get from you can get the sequence from anywhere. Okay, so you have to paste your amino acid sequence. So if you know that Uniport ID or a Trimble ID, just provide the Uniport Trimble ID here. Okay, click on compute parameters. It won't take much time for you. It will provide you results for you. So here in first column it will give you what exactly the information you provided. So you can see here we provided this sequence, the total number of now we have, so you're going to get the results here. So it will show you total number of amino acids. You can see here we have 631 amino acids are there. So molecular weight, total molecular weight of your protein, and this theoretical isolated point, it is 8.69. Okay, and also you can see here composition of each and every amino acid in a protein. So alanine, 28 residues are there, nearly 4.4% is covered throughout the protein. So total number of charged residues, you can see here aspartic acid, glutamic acid, acetic acid residues are 83 are there. The basic residues are 93 are there. Arginine and lysines are there. Okay, so and also you can see the atomic composition also, with total number of how many atoms each and every atom has been covered throughout this protein. And also it will show you a molecular formula and also total number of atoms. So overall total number of atoms are there, it will show you. Along with this, it will show you a very good parameter such as the extinction coefficient. Okay, so this extinction coefficient indicate how much light a protein can absorb at certain wavelength. Okay, this is very important. So when you're reading the when you're working on the protein kinetics, sorry, enzyme kinetics, it is highly essential, highly helpful for you to check the wavelength, how much light protein has been absorbed in a certain wavelengths. So also it will show you a half-life also. So half-life means the half-life is prediction of the time it take for the amount of protein in the cell is disappear after it senses in the cell. The prediction has been given for the three organisms. You can see here it has given for the three organisms. So generally the protopram, this software estimate based upon the N-terminal amino acid sequence. Okay, based on N-terminal amino acid sequence, it will give you what is the half-life of this protein or enzyme will be there. You can see here the half-life of this enzyme or protein, 30 hours in the mammalian. It is compared to it is more compared to the yeast and the other organisms. Okay, and also it will give you a instability index. This instability index is it will provide uh, stability of your protein in the test tube. So what is the stability index? What is the stability of your protein when it has been taken into the test tube? So if the value is less than 40, if the value is less than 40, it is stable. Okay, so if it is value is more than 40, it is unstable. In this case, if you take this protein in a test tube, it is completely unstable. Okay, it's not going to give any accurate results for you. So the last parameter also, it will show you aliphatic index. What is this aliphatic index mean? It's the relative volume occupied by the aliphatic side chains like alanine, valine, isoleucine, and leucine. Okay, so actually it may regards as a positive factor for increase the thermostability of globular proteins. Okay, so this is called as aliphatic index. It will provide an aliphatic index for you for how much index it has, and also the last one, it will show you a grand average of hydropathicity, GRABI we call this one. So this value of all amino acid, what is exactly this mean? That it's a value of all amino acid divided by the number of residues in a sequence. So it will provide you how much hydrophobicity hydrophobicity is there in your protein sequence. Like this, it will provide you basic information, basic physiochemical properties for you if you provide a sequence. It's a very good tool if you are working on the if you are characterizing your protein. You can use all this information for your papers or publications. Okay, so this is about the protoform, very brief about the protoform. So next we will see about one more tool. We call that one as a peptra. So it's almost identical, but it has its own limitation. For the physicochemical properties, you better to use this protoform only. So next one, we are going to see about a tool, peptra. Okay, so here this is a peptra. Here you have to provide a sequence, or you have a chance to draw your own sequence. Okay, for example, if I want to draw a sequence like this, so you can see here I can easily draw a sequence. Okay, you have to click on draw peptide. So one good thing is with this Peptra is it will generate a two-dimensional figure for you. Along with that, it will show you some of the basic parameters such as isoelectric point, net charge, hydrophobicity, extinction coefficient. Okay, so length, what is the mass of this peptide? It will show you information for you. So the major disadvantage with this Peptra is you can draw up to 60 amino acids only. Okay, so you can use a maximum of 60 amino acid length of peptide only. You cannot go further more. So that's a major disadvantage with this one. If you want to draw a peptide, a small peptide, it is highly useful, highly uh, flexible for you to save the two-dimensional images along with some of the basic parameters. So but as I told you, if you are checking the physicochemical properties, better to use this program XPASIC tool only. Okay, this is about the first tool. Now let's go back, let's check the second tool, prediction of domains in the proteins. So here we have three different tools are there. So one is Interpro, and second one is Smart, and also the PFAM. Okay, these are the three various tools we are going to use. Okay, so we are going to use to identify the domains of the protein. Okay, first let us check with the Interpro. So let's give me some basic introduction about the domain. Domains are the functional and the structural unit in a protein. So usually they are responsible for the particular function, or they may be involved in the interacting with the other protein. Okay, so overall, generally they will carry out the overall role, major role in a protein or enzyme. Okay, so let us see how we can identify the domains in a protein, in a unknown protein. Okay, so let us go back to the tool. We are going to check three different tools. One is Interpro, Smart, and PFAM. Okay, so here I'm going to close this one. So Interpro. Okay, so it's EMBL1. So here very simple one. It's also not going to be any complicated things for you. So you have to provide only a single letter amino acid sequence. Okay, so here I copied the sequence, the same sequence I'm going to use. So here paste. Okay, so just give me some basic introduction about the Interpro. Interpro is a resource that provides a function analysis of a protein sequence by classify them into a families and predict the presence of domains and important sites. Okay, so Interpro is always is the best one to check the domain parts. So if you are working on the domain and domain analysis, always as preferable to use this Interpro. Why means it generally use the predictive models known as the signatures. Okay, uh, these signatures is provided by the various database like a different database that makes the Interpro consortium. Okay, so this is very unique compared to other databases out there like a PFAM, Smart. So why it is unique mean it is a combination of various databases. It's not a one particular database. It consists of a nearly 14 databases out there in Interpro. That's why we always prefer to use Interpro even in our research also. Okay, so if you want to work on the domain part. So here I pasted the uh, sequence. Click on search. So it will take almost 2 to 3 minutes to get the results of this your sequence. Okay, once it is done, it will show you a option like this job has been finished. You just need to click this link. So you can see here it will show you a beautiful figures for you, various domain along with the various databases. Okay, you can see here this SH2 domain predicted by the Interpro domain from the region of 245 to 345 amino acids. So if you scroll down a little bit more, it will show you here smart database has predicted from similarly is almost identical with the Interpro. It's predicted as 245 to 336 domain. So if we go to a little bit more here, you can see here the PFAM, the prosite profile, and also you can see the PFAM. So like this, it will predict a various database has been involved here. It will show you various kind of results for you. Along with this, it's also show you active sites, uh, active sites also for you, binding site, what are the conserved regions are there in the protein, and what are the super families, homologous super families are there in the protein by in various by comparing with the various databases. So there is a few more options are available for you. Okay, if you can download this whole image in a single shot. So if you want to check only a specific database, that's also option is available. Click here on entry. Now you can see here you can select whatever the database you want. For example, if you want only a PFAM, so here you can select the PFAM. Now you can see here the 5 different domains are there in the PFAM. Okay, so the first domain here, it's a protein kinase tyrosine serine 3 domain. It is started from 370 to 618 amino acids. Similarly, you can see here, so one more domain is a BTK domain and PH domain, SS3 domain, SS2 domain. Like this, 5 domains has been identified in a PFAM. Okay, if you want to look onto the only Interpro, you just go for it. You can see here the 14 different domains it has been predicted. Like this, you can easily understand or predict the domain information by using the Interpro. Why it is unique means, so it has a combination of 14 different databases are there uh, in an Interpro. That's why we always prefer to use this Interpro prediction for the domain analysis. Okay, so let us see one more database. So smart SMART. Okay, this is a EMBL tool, EMBL smart EMBL. So here type smart EMBL, it will show you a main web page here. So here in this database you have to provide only a sequence information here, or you can give the accession code. If you already know the accession code, you can provide accession code, or you have to provide only a sequence. So here I provided a sequence only. So already a known sequence of our complementary C1S1 C1S. Click on sequence smart. So you can see here it is going to provide you a beautiful graph, a graph or domain regions for you in your protein. Okay, you can see here there are 1, 2, 3, 4, 5 domains are there, which has been predicted by this smart. It is also very identical to that the thing what have we have shown in Interpro. It is also showing a similar kind of results for you. See here, it's a pH domain, BTK domain, SH3 domain, SH2 domain, and also tyrosine Kc domain. And also here it will show you where exactly the start and stop of the domain along with the expected value. So the value is always in negative. If it is significant, if it is less than 0, it means it's highly significant. Okay, so and also it will provide the basic information also for you. If you click this particular domain, it will show you what exactly the function of this domain. So based upon the function of the domain, based upon the role of the domain, we can easily predict what exactly the function of this particular protein is. So as I told you previously, domains are the functional and structural of the life. They determine what exactly the role of that particular protein. Okay, so here in information, if you provide you a basic information about this protein, if you go to architecture, it will provide you a domain architecture, how exactly it looks like. So if you go to interaction, it will jump into the string database. So how, what are the other protein has been interacted with this your gene or protein, it will show you. If you go to pathway, it will show you what are the pathways are there, what are the major pathways are there in the protein. It will show, you can see here, it play a very important role in T cell receptor signaling pathway. And also if you go to orthology, it's a similar organism, what exactly the role of this protein in other species, it will show, you can see here, mostly it is a tyrosine kinase. Okay, so it's the enzyme activity, it's one of the enzyme called as a tyrosine kinase. So we can from this analysis we can predict that our enzyme is nothing but a serine tyrosine kinase, which play a very important role in various kinase reactions. Okay, so like this, based upon the domain, we can, it's easy for us to predict what exactly the function of the protein. So you can save this image, it's very easy. So here there is option is available for you. You have to click on the save. Now we can save this image in a SVG format, where you can use Photoshop to create that one is a JPG format. Okay, this is about the smart. It's almost identical to that Interpro, but it will give you more information for you, like architecture, interaction, pathways, PTM, and orthology. Okay, so let's check one more database on the same one, on the same string, the PFAM. Okay, so this is the most popular database. Most of the researchers prefer to use the PFAM database, but I will suggest you to use the Interpro and Smart if you want to represent in a very good graphical representation. This is also a very good database, no doubt about it. Okay, so maintained by the same EMBL and eBay centers. Okay, so here as input you can give anything like the ID number or keywords. So, okay, so your sequence, anything you can provide as input. So we are going to give same sequence. Okay, just go for it. So you can see here it's also provide the same similar kind of information what we have seen in Interpro. So and also in a smart, 5 different domains it has been provided. Okay, so it will show you graphical representation also for you, what is where exactly your domain has been located. So and also it will show you a start and end amino acid. Okay, so and also it will search for the look for the domain region alignment. If you click on this one, it will show you the alignment region uh, with your sequence, here, and this is the sequence which is got it from the databases. So basically this PFAM runs with the hidden Markov model statistics. Okay, so we all know the hidden Markov model is a statistical model that can be used to describe the evolution of detectable events. So that depends upon the internal factor. which are not directly visible for us. Okay, so we are using, generally the PFAM uses the HMM model to predict the domains. Okay, so here you can see here the starting from the HMML prediction, starting from 1 to 258 base pairs, it has been found a match with the already available domain in the database. This is our sequence, it's already available information in the database. So if you click this protein, this domain, it will provide you information what exactly the role of this protein. Okay, so if you click here the PFAM, it will show you what exact the role of this protein tyrosine kinase. Okay, and also it is linked with the interpro with this database. So much of information. If you want to check each and every information, there is so much of information available regarding that particular domain. You particularly click only one domain, it will show you so much of information for you. It may be helpful for your uh, if you're working with the specific domain, it might be helpful for you to find out what exactly the function of your protein is. Okay, so like this, these three databases are vital. If you're working specifically with the unknown proteins, it may be helpful for you to find out what exactly the function of your protein by comparing the three different annotations in three different databases. One annotation in three different databases, it will give a similar kind of results for you. It will give strong support for you to interpret what exactly the function of your protein is. Okay, this is about a protein domain. Now we are going to look onto the one more annotation, how we can identify the transmembrane region. So there are, if you are working with that, if you are working with the membrane protein, it is very vital for us to understand uh, how exactly that membrane has been spanned in the lipid layer. Okay, so the transmembrane prediction predicts the membrane spanning region and their orientation. Okay, generally the algorithm, so here the two different databases are there, two different tools are there, THMM server and TMPRED server. So these are the most popular server to used to predict the transmembrane region. So generally they use the algorithm based on already available information on naturally occurring transmembrane proteins. So the prediction is made. The prediction is made using the combination of several weight matrix scoring methods. Weight matrix scoring method. They use this weight matrix scoring method to calculate or to evaluate how exactly transmembrane has been embedded uh, in a lipid layer. Okay, so it will show you uh, what particular region is outside of the region, outside of the transmembrane, what particular region is spanning inside the lipid layer, what is the region which is present in the cytosolic region. It will show you information for you. This is vital for us if you are working on the transmembrane. Why means from this analysis it will show you how exactly the ligand should be bind with the receptor molecule in the exterior or in interior environment. Okay, let us start. First, we'll check about TMHM server. Yeah, so here we have to provide the amino acid sequence, identically similar thing. So I'm going to use this hydroxytryptophan receptor 2A. Okay, this is one of the receptor. So here I'm going to paste this one. Okay, so here click on submit. So now it will provide the results for you. You can see here the total number of amino acid. It will show you 471, and also so here you just need to provide the amino acid sequence, and you need to keep everything as default. So if you want a graphic, you have to add this one. If you don't want any graphic, just click on the no graphics. Okay. So here As a result, first term we are going to get the total number of amino acid, and also you can see number of prediction. It will show you seven transmembrane has been predicted. Okay, so you can see here this region. Okay, so here this region is belongs to the transmembrane. 1 2 3 4 5 1 2 3 4 5 6 7 seven helices this is the exterior one. Okay, this is the exterior one, I mean the outside of the transmembrane, and this is a membrane protein. So this is a spanning in the membrane. Okay, this region has been spanned in the lipid membrane, and this part is belongs to the thick part has been belongs to the inside of the cell. Okay, inside of the cell, it will show you your location exactly. That you can see here the outside of the cell, starting from first amino acid to 76 amino acid, it has been located in the outside, and you can see 77 to 99 amino acid, it has been located in a membrane spanning. You can see 100 to, there is a loop. Okay, there is a connector in between these two helices. You will find 100 to 111 amino acids in the inside of this. Like this, you can easily identify the transmembrane region in your protein sequence. Okay, so along with this one more database is there to predict the transmembrane region. Okay, this is called as tmpred. So here tmpred. Okay, so tmpred, so it's also identical to that of previous database. Okay, so here previous annotation tool. So click on, you have to paste your amino acid sequence, very simple. Okay, you keep everything as a default, click on run trap. Now it will show you a same information as we have done with the previous one. You can see here 7 outside helices has been found. It will show you same kind of location, similar kind of location for you, same kind of result for you. You can see here possibly transmembrane helices. Okay, so the location, inside and outside regions based on the amino acid length. Along with this, all this same similar kind of information, it also provides you a hydrophobic plot. Okay, so this hydrophobic plot will, what it exactly will show you means here, this is the x-axis that represents the amino acid. Okay, these are the amino acid, and the area above zero. Okay, so here the area above zero indicate the hydrophobic portion of a protein. Okay, so the area above determine the hydrophobic portion of the protein. Those are under zero, the hydrophilic portion of the hydrophilic portion of the protein. So it shows that there is a almost a 1 2 3 4 5 6 7. You can see here as it is seven possible membrane spanning regions are there in the protein. Okay, it's a 7 transmembrane helices. From this analysis we can predict that it's a 7 transmembrane helices. So you can save this information. If you want, you can just click, like, need to click this one to right click save image. You can save this image also. So like this, we can easily predict the transmembrane region by using this temperate one. So when you see the comparison between this tool, almost they are identical, but it will show you a hydrophobic plot here. There it will show you in a previous tool, it will show you directly a graph which is representing the transmembrane region in a protein.